This presentation is about bacterial structure. The key questions are, what structural features do bacteria have, and how are bacteria adapted to their functions? Their classification is of prokaryotic cells, meaning they have no true nucleus. They are the most common life form. Some bacteria are pathogenic, causing disease, whereas some bacteria can actually be beneficial to us. The different shapes of bacteria can be used in a classification system. Bacteria can be seen with a light microscope, as they're only about 2 micrometers in length. By using their shape, we can identify what type of bacteria they may be. The bacilli, the cocci, spirilla, and vibrio are different types of shape for bacteria. Bacilli are rod-shaped, whereas cocci are spherical. Spirilla are spiral-shaped, and vibrio are common-shaped. Pleomorphy describes how bacteria can change their shape due to their age or environmental shock, for example, changes in temperature. This can make their identification due to their shape quite difficult. These are the common features between different bacterial structures. They all have rigid cell walls made of peptidoglycan. Peptidoglycan is a polysaccharide with amino acids adjoined to it, and its alternative names are mucopeptide or murane, from the Latin murus, meaning a wall. Bacteria have plasma membranes which are similar to eukaryotes, and they contain cytoplasm, which is the site of chemical reactions that take place in the cell, and this cytoplasm is held in one compartment which is formed by the plasma membrane surrounding it. Now, the DNA is a double-stranded loop of DNA, which we call the bacterial chromosome, and it's in a condensed area called a nucleoid. Notice that it's not a nucleus because there's no membrane or nuclear membrane surrounding this genetic information. There are small 70S ribosomes, which are involved in protein synthesis, and for energy there are storage granules made up of glycogen or lipid droplets. Finally, there are plasmids, which are smaller rings of DNA. Now, these are of specific interest to genetic engineers because they can convey genetic information between bacteria, and we can actually harness that in order to genetically modify other organisms. Now, here are some additional bacterial structures that aren't found in all bacteria. Some bacteria are motile, which means that they can swim or they can move. They have thin fibers that are called flagella. These are corkscrew-shaped and rotate, which propel the bacteria in different directions. Other fibers, called pili, enable the bacteria to perform a primitive form of sex called conjugation. Conjugation simply means joining, and during conjugation the pili become joined and form a channel to allow the plasmids to be copied and transferred to other individual bacteria. Mesosomes are where the cell membrane folds inwards and is present in gram-positive bacteria. We'll talk about this a bit more later. The mesosome is the site where many enzymes are found, including respiration enzymes for energy release. The mesosomes will increase the surface area for metabolic activity, and they're probably involved in cell wall synthesis and cell division. Some mesosomes may be present in some gram-negative bacteria, but are smaller. And in photosynthetic bacteria, the membrane is folded to form the thylakoid membrane. Some types of bacteria also have a capsule, and this is a sticky coat outside the cell wall that prevents the bacterium from drying out, from being digested by host intestinal enzymes, or from being attacked by the host's own immune system. So here is the typical diagram you would find of bacterial structure. The labels that are marked with an asterisk are those that aren't found in all bacteria. So it's important that you go through this and you learn how to label a bacterium like this. You may want to pause this video and take a note of this. Okay, so here's another specific example of an E. coli bacteria, and you can see that there are all these common features such as the plasma membrane, the cytoplasm, the cell wall, the DNA, and the ribosomes. But we find the extras like the flagella or the pili on the surface of the cell. So here's some more information about bacterial cell walls. Bacterial cell walls are made of peptidoglycan, which are hexosugars and amino acids. The alternative names are mucopeptide or murine, depending on which textbook you're reading. And cell walls are strong and flexible. They support the cell and protect the cell contents, either from drying out, from being attacked by enzymes, or by the immune system of the host organism. Additional information includes the fact that some drugs and disinfectants destroy or prevent the synthesis of peptidoglycan. Antibiotics work in this way in the fact they interfere with the synthesis of cell walls of bacteria, and antiseptics can also cause bacteria to split open on surfaces by interfering with the peptidoglycan structure. Your tears naturally have enzymes in them called lysozymes, which hydrolyze the peptidoglycan, therefore killing the bacteria. So you may have already heard about gram-positive or gram-negative bacteria. So this is a method used to classify bacteria based on the result of us trying to stain bacteria for sight on a microscope slide. So gram-positive bacteria have thick peptidoglycan walls, and so they thickened walls with additional polysaccharides and proteins will actually hold a stain called crystal violet stain. So examples are Bacillus, Lactobacillus, and most cocci species, such as Staphylococcus. Those bacteria are gram-positive bacteria with thick cell walls. Alternatively, the gram-negative bacteria will have thinner walls with less peptidoglycan in them. They have thinner walls with a surface layer of lipids instead for protection. These, however, do not retain crystal violet stain after you've washed them with alcohol, so we would use another stain to counter-stain them, called safranin. And examples of gram-negative bacteria with thinner walls are E. coli, Salmonella, and some other cocci species. So just to recap, gram staining of cell walls can help us to identify the type of bacteria, and gram staining can result in a gram-positive or a gram-negative result. And gram-positive bacteria have walls that are thickened with additional polysaccharides and proteins, so they retain crystal violet stain, whereas gram-negative bacteria have thinner walls but with a surface layer of lipids for protection. And because they have thinner walls that don't take up this crystal violet stain, you use another stain called safranin to counter-stain. So let's take a look at gram-positive bacteria cell walls. In the diagram you can see that the peptidoglycan layer is actually really thick, and because it's so thick it retains this crystal violet colour so that it stains a dark purple colour. Opposite to this, we have gram-negative bacteria. Gram-negative bacteria have thinner cell walls with less peptidoglycan in them. Because they have less peptidoglycan, they don't retain that crystal violet stain, so you have to treat it with a counter stain, another type of stain called safranin, and therefore the safranin is like a light pinky colour as opposed to a dark colour. So if it becomes a dark colour, it's the gram-positive. If it's like a pinky light colour, then those are your gram-negative bacteria, and it's due to the amount of peptidoglycan and the thickness of the cell wall. So the cell wall structure of gram-positive and gram-negative bacteria can be summarised in the following table. And you can see the total thickness of the gram-negative bacteria is a lot thinner than the gram-positive bacteria. The thickness of the peptidoglycan within it is 20 to 80 nanometers in the gram-positive bacteria, whereas it's only 1 to 2 nanometers in the gram-negative bacteria. So in addition to the peptidoglycan, we know that the surface membrane with protein and lipopolysaccharides are not present in the gram-positive bacteria, whereas they are present in the gram-negative bacteria. And so they're using this lipopolysaccharide as not only protection, but attachment as well. In terms of gram-positive, gram-negative, there is a compound called teichoic acid in the membrane of gram-positive bacteria, but not in gram-negative bacteria. And teichoic acid in this membrane will help gram-positive bacteria to regulate their shape and also the rate of their cell division, among other things. Also, among the things that are on some bacteria but not all bacteria, we could have capsules and slime layers. They are outer surface coverings. Now, it's important to note that the slime layers sit on top of the cell walls, and the cell walls sit on top of the cell membrane. So all bacteria have cell membranes and cell walls, but not all bacteria have slime layers or capsules. Now, the slime layer is loose, it's soluble, and it's easily washed off. In terms of a capsule, they tend to be thicker, and they're made of polysaccharides and or polypeptides as well. Now, these coverings, the slime layer and the capsules, will reduce the drying out, the desiccation process. They cause the cells to stick together, and they prevent antibodies and phagocytes from your immune system from binding with the cell wall. So they're a method of protecting them for long enough so that the bacteria can multiply within the capsule and cause an infection. The other optional extras of bacterial cells are flagellum, pili, and fimbriae. So the flagellum are found in some bacteria. They're used for rotational movement, and that rotational movement will help the bacteria to be motile or able to move to get to where it needs to go. And they're made of the protein flagellin. The pili and the fimbriae are sometimes present, and they project from the surface membrane through the cell walls. These will help attachment to other bacterial cells or to host cells when they're interacting in an antigenic way. They're causing a takeover effect. A large number of fimbriae may hinder the phagocytosis process by the host cells, so it serves as a protection function. So attachment and protection are very important here. Whereas some pili are sex pili, and they're associated with this conjugation or this joining process where bacteria will form conjugation tubes and swap copied DNA. Bacteria reproduce in a process called binary fission, which literally means splitting into two. It's the most common form of reproduction, and a division process can happen every 20 minutes, making the rate incredibly quick, and the rate of growth can actually be exponential. In this process, circular chromosomal DNA is broken open by enzymes, and then the DNA can replicate. The new cell wall and the membrane will divide the cell into two new parts, and so this process is actually very simple. Genetic material can be exchanged in three different ways. Transformation, where DNA is released by a donor, taken up by a recipient to replace similar pieces of DNA. It can be transduced, where a small amount of DNA is transferred by a bacteriophage, so it's a virus infecting. During conjugation, the donor cell contains a plasmid that contains the DNA that will code for a formation of a sex pilus, so that the information can be exchanged. Now try a question about prokaryotes. Pause the video and then unpause to see the answer. Now try this checkpoint question. Remember to write your answer out in full. This concludes our presentation about bacterial structure.